Hello, everyone, and welcome to today's Gen webinar, which is entitled, Are Your AAVs Filled? Determine Rapidly with Mass Photometry. Our webinar today was made possible through sponsorship from Refine, Measuring Molecules with Light. I'll be your host for today's event. I'm Jeff Bogaliskas, Technical Editor for Genetic Engineering and Biotechnology News, which has been bringing you the latest information about innovations within the life sciences industries for over 40 years. Now, the continued success of gene therapy research has placed a demand on the biomanufacturing industry to produce the go-to therapeutic delivery molecules, which for so many gene therapy endeavors is adeno-associated virus, or AAV. Many of you already know about AAV and its role in delivering gene therapy products, as it's been widely studied for a number of years. However, what many of you may not be aware of are some of the challenges associated with manufacturing AAV at scale, in particular, quantifying the ratio of empty and full AAV capsids. Currently, most existing methods are not quick and simple enough to be employed throughout the manufacturing process, a scenario many scientists would like to rectify. Well, in today's webinar, we're going to learn about mass photometry technology and how it's being tailored to address the challenges of AAV characterization. Here today to tell us more about mass photometry is our presenter, Dr. Svea Cheeseman, who is a product manager at Refeyn, responsible for products targeted at the biopharma industry. Svea has a background in single molecule biophysics and a very keen interest in pushing the boundaries of biomolecular analysis to eventually enable real-time release of biomolecular drugs. Now today, she is going to tell us more about Refeyn's novel mass photometry instrument, as well as provide data providing its ability to reliably quantify the empty-full particle ratio for different AAV serotypes and at different purification levels. Now, before we get the webinar underway, I want to remind the audience that we'd love to hear from you in our Q&A session that will begin right after the presentation ends. Svea will be on hand to try to answer as many of your questions as she can, so simply type your questions into the Q&A box and hit submit. You can do this at any time during the presentation. You don't have to wait until the very end. So if you have a question that pops in your head, type it on in and send it on away. All right, enough from me now. I'll hand things over to Svea.
Hi, everyone. Thank you so much for joining our webinar today. Are your AAVs filled? Determine rapidly with mass photometry. My name is Svea Cheeseman. I am a product manager at Refine, responsible for the biopharma market. And today I'd like to speak about the adeno-associated virus, AAVs, and more precisely about the question whether they are filled, which means that they have the genetic material with the transgene they're supposed to deliver, or whether they are empty, which means that they have no DNA load and are just empty shells. One of the biggest challenges in the production of AAVs nowadays is that many processes end up with a high amount of empty particles. And this can pose several risks. Firstly, it leads to a low transduction efficiency due to receptor binding competition. It leads to increased immune responses. It limits the target genome concentration due to capsid precipitation, and finally results in increased cost of goods produced. Now, we think that one of the major reasons why there are so many empty particles, even in manufacturing, is that there's a lack of a technology that can quickly and reliably assess the empty-full ratio early in the process. So what would such a solution look like? It should accurately determine the empty-full ratio within minutes, should ideally be able to quantify partially filled particles. It should use a low sample volume and particle concentration so that it can be used early in the process with little virus around. It should be easy to use at low operational cost, agnostic to different serotypes, and also applicable at different purification stages throughout the process. And we think that mass photometry could be a solution to this problem. So let me quickly explain how mass photometry works. We have here a microscopy glass slide and your AAVs in solution on top of the glass slide. Now we shine light on it from the bottom, and what we capture is the interference of scattered light and reflected light. The reflected light is always the same, reflected by the glass surface, and the scattered light comes from scattering molecules in the solution. And the extent of scattering depends on two things. One is the size of the scattering object, the AAV in this case, and the second is its refractive index, so its optical density, which both of these then relate to the mass of the molecule. So what we see is the interference of the scattered light and the reflected light that then look like these points that we capture on a camera. Every single point corresponds to one single AAV molecule that we detect. Now, interestingly, the more the molecule scatters, the darker this inner circle is, so the higher the contrast on the camera. And the lighter the molecule, the less contrast we see on the camera. So we analyze the contrast to then get these histograms of different molecules, and we can transfer the contrast, translate that into molecular mass using a calibrant. Now, this technology is very well applicable for the problem of empty versus full AAVs. That is because there is a significant mass difference between an empty and a full AAV. Empty AAVs typically weigh around 3.7 megadalton, and full AAVs are typically around a megadalton heavier than that because of the DNA load. Also, what is nice for our technology is that contaminants are typically of a much lower weight, below 2 megadalton. So they also don't interfere with the measurement. And now when we put our AAVs into our mass photometer, a typical result can be seen here at the bottom, where you get the single molecules counted and their mass over their mass. And you see that we get two distinct peaks, one for empty AAVs and the heavier one for the full AAVs that are baseline resolved. I'd like to quickly show you this video to show you how easy it is to use mass photometry in your lab. So basically here you see this glass slide, and now a sample is applied without any labeling, just a pure sample on the scope. Now you see here these dots that I explained to you earlier. These are single molecules landing on the surface, and we record a movie to capture those molecules. In the next step, the movie is transferred to a data analysis software, Discover MP. And this data analysis software now identifies every single molecule and saves the information on the contrast. It then gives you a histogram of the counts over the contrast. You can load a calibration sample that you ran earlier. It's just different plotting with different masses. And then you can get the mass histogram. So it's a very easy to use technology. Now, I'm very excited to introduce to you a new mass photometer that we are bringing to the market that is called the Samux MP. And this is a dedicated mass photometer for the use with AAVs. So we changed the mass photometer in three significant ways to receive the Samux MP. Firstly, we optimized the technology to a mass range relevant for AAVs, which is between 3 and 6 megadalton. We also changed and optimized the hardware so that it can easily be decontaminated and doesn't pose a risk of remaining AAVs. Finally, we changed the software to optimize it for a quick and easy assessment of empty-full ratios. Now I'd like to show you some data on how this instrument performs with AAVs. So let me start off with the experiment showing the range of empty versus full particles. The data on the left was acquired in our own lab, where they used 5 different mixtures of empty and full particles that we commercially purchased. ranging from 0 to 95% full. And you can see that we could measure the loading rate with a very high correlation of 0.996, showing that we can reliably measure throughout the entire range. One of our early partners, Cell and Gene Therapy Catapult, also did a very similar experiment, but they used their own preparation of an empty AAV particle and full preparation of 70% full AAVs. And you can see in this graph that they also saw a really nice correlation throughout the measurement range, but they also saw a very big variability of the measurement, especially when there was a high mixture rate of 50-50 of the two preparations. Now they thought that this could be introduced to the mixing rather than through the variability of the Samux. So they next performed the following experiment. They wanted to assess the precision and reproducibility of the Samux MP, and for that, they produced 6 independent replicates and measured each 6 times by 2 different operators. And then here you can see the data plotted that was received, and from that, the repeatability and the intermediate precision was calculated. You can also see the acceptance criteria for the qualification by Catapult. The repeatability was supposed to be below 20%, whereas the precision should be below 30%. And you can see that the results of the Samux MP are much better than that, with around 11%. So the Samux MP passed the internal qualification in terms of precision and reproducibility of Cell and Gene Therapy Catapult. The next thing we wanted to test is whether mass photometry is truly agnostic to different serotypes. The data on the left is acquired in our own lab with 4 different serotypes commercially available. And you can see that they all give a similar peak in resolution. And I want to stress that the sample preparation and the sample analysis was the same for all of these 4 samples. So there was no difference seen for different serotypes. Another partner of ours, the Utrecht University in the Netherlands, they did a very similar experiment where they used 2 different AAV8s from 2 different suppliers and also an AAV5, and they saw the same result, that there's no difference between different serotypes. But maybe even more interestingly, this lab of Professor Albert Heck, they are experts in charge detection mass spectroscopy. And they used one of the AAV8s and put that into the mass spectrometer to compare the mass that results from the mass spectrometer with the mass determined by the Samux MP. And you can see that at 3.83 versus 3.81, it's a really nice agreement showing that the Samux MP also reliably determines the mass of the AAVs. We further extended this benchmarking study. The data on the top again are from Cell and Gene Therapy Catapult. They measured 4 different samples with 6 to 60% full particles, with the 2 gold standards for determining the empty-full ratio, cryo-TEM and analytical ultracentrifugation AUC, and compared that with the results received on the Samux MP. And you can see that this data is in great agreement, showing the reliability of the method of the Samux MP. Pharmaron, another partner of ours, they also measured the empty-full ratio with cryo-TEM, IUC, and the Samux MP at their plant in Liverpool. Unfortunately, we couldn't get the TEM data for one of the samples, but they had a 16% full sample and a 100% full sample, and have AUC and TEM data on that. And you can see that the Samux MP results, again, nicely agree with what they have measured with the gold standards. So I hope I could show you that the Samux MP reliably measures the empty-full ratio of AAV. However, the instrument can do much more than that. And I'd like to stress that there's very little sample preparation required. This is a label-free measurement in native state, and it doesn't require much sample preparation. Also, results can be obtained really quickly. Typically, a measurement takes less than 5 minutes, including the analysis step. We also use very low sample amounts as we are a single molecule technique. We typically use 10 to 20 microliter of 10 to 11 particles per milliliter. This is where we receive the best results. It is very easy to use. New operators are typically trained in less than a day. It is a benchtop instrument and therefore has a very small footprint. And finally, a measurement costs less than $5, depending on the calibrant you're using, which also results in very low operational costs. And I'd like to drill down a little bit more into the ease of use of the system and show you a screenshot of our analysis software, Discover MP. So this is what it looks like once you have analyzed a movie of your AAVs. And what the user does is they determine the range for the empty and for the full. This can either be done by dragging it with the cursor, or you can also manually enter it down here in this field. Once you have done that, there's an automatic analysis given out that you see here. So it automatically gives you the amount of full capsids, the single molecule count and percentage, the empty capsids, and the ambiguous capsid mass, so those molecules that were not inside these ranges. And finally, the full proportion. Something that our users really appreciate is that the analysis software also has this figure design tool where you can load your data into this figure, and you can change it according to your preferences. You can change the labels, and then this helps you can directly export it into a PowerPoint or report. So this really allows for quick and easy reporting. We also supply consumables with our mass photometers. So you don't really need a lot of consumables. Mainly it is the sample carrier slide that you see here and the sample well cassette. So the silicon mask that gives you different wells to apply your samples. These are available from Refine. So that allows you to even further shorten the preparation time and gives you more confidence in your data because the conditions are always the same. One last sneak peek I'd like to share with you. We are going to introduce more additions to our mass spectrometry lineup other than the Samux MP. One of them is this automated mass spectrometry that you can see on the left. It's a robot that you can fit on mass spectrometers so that you don't have to manually apply samples anymore. Stay tuned for the market communication of this system. You can get in touch with us through our website or social media. And with that, I'd like to conclude the presentation. I hope that you appreciate that Samux MP precisely measures the empty-full ratio within minutes for any serotype at minimal operational costs, that it uses low sample volumes and concentrations, and very little sample preparation is required. And finally, it's an easy experimental procedure with a tailored data analysis software. The Samux MP is now available for demonstration, so please contact us if you're interested in this machine. With that, I'd like to close my presentation, and I'm very happy to take your questions.
Svea, really great presentation and a pretty amazing device. I'm sure the audience has a lot more questions for you about your data and your presentation. So let's move along to the Q&A session and try to tackle some questions, shall we? If you have a question for Svea, don't hesitate to send it on in, and bear with us for just a moment as we make the transition into the Q&A session.
All right, thanks everyone for joining us for this Q&A session. Svea is on hand. You guys have some excellent questions that have been coming in. Keep them coming. We're going to try to get through as many of them as possible. So Svea, we start our first question. One of our audience members would like to know, what is the mass range that the Samux MP can measure?
Thank you for the question. The mass range is from 500 kilodalton to 6 megadalton. Likely we can go a little higher than that. We have to be very honest with you, not determine the exact upper range because we are lacking a sample that is that big that we can measure, but it certainly can measure between 500 kilodalton and 6 megadalton.
All righty, thank you for that. Next question we have is, which kind of purification AAV productions have you tested? Was it crude lysate? Was it ionoxanol purified column?
So with the development partners that we've shown the data from, I cannot tell you because they haven't disclosed that. I can tell you that the samples that they used for the comparison with AUC and TEM, they were purified in the identical way. So they used identical purified AAVs for all three different methods.
All right, thanks. Next question we have, this is a good one. Could you tell us a little bit more about the calibration sample, about what it is, perhaps?
Sure. So basically, for calibration, you just need a protein of a known mass that is within the measurement range of the mass photometer, so between 500 kilodalton and 6 megadalton. We typically recommend at least a 2-point calibration. So what we can recommend in terms of what's available commercially is thyroglobulin, which gives you a peak at 670 kilodalton, and then add an empty AAV to that of which you know the mass, and then typically this is good enough to calibrate the system for mass.
All right, thank you for that. Next question we have is, and I just want to let you know, can the MP resolve partially filled AAVs and be able to determine partial genome incorporation from the empty and full distributions?
Yeah, so that's an interesting question. We are pretty confident at this point that we can do that. And you may have seen in some of the graphs, the histograms that we've shown, is that there's a full and an empty peak, and then there's something in between. So it doesn't go all the way to zero. There is particles detected, and these particles are partially filled AAVs. We're currently still investigating together with partners to validate that we can reliably quantify the partially filled capsid ratio, but we are very confident that we will soon be able to launch that as a feature.
All right, thank you. And how does the system handle 3D aggregates of AAV?
So with aggregates, we do see them. It's a bit of the same story as with the partials. We do see aggregates. We are currently investigating how well we can quantify them. It also depends a little bit on their size, how big they are. But you can see them in the mass histogram.
All right, thank you for that. So the next question an audience member has is, do refractive index differ between empty, say protein, intermediate, and full protein and DNA together?
Yes, that is the very basic of the technology. So there's a change in the refractive index, hence we see a change in the contrast on our camera, and therefore we can detect the difference in mass. That's correct.
All right. And another audience member would like to know, what was the purity of the preps in comparison with cryo-EM?
As I mentioned earlier, so these were prepped in the same way for the benchmark comparison. We have also tried different purification stages. The power of mass photometry is also a bit what lets it down sometimes, is that we see every single protein. Right? We see everything that's in the mixture. So if you take a crude extract, you will see everything that's inside on the mass photometer. That means that your resolution will go down, especially if you have something as big as an AAV in there. So it really depends on what resolution you need and what it is that you want to get out of the measurement. That determines how much you have to purify your sample.
All right, thank you for that. Here's a pretty good question, I think. An audience member would like to know, can mass photometry be used to quantify the virus titer?
So that is an interesting one. We cannot at this point quantify a concentration of molecules. So with relative concentration, we are quantitative, but with absolute concentrations, we are not at this point. The main reason is that we are very, very sensitive in terms of single molecule counting. And we apply the sample manually onto the sample stage. and there is a certain variability in the procedure, like how it's pipetted, how much exactly, with what speed, at what time is the movie started. And this variability means that we are not reproducible in terms of counts to an extent that we can reliably measure the concentration. However, we think that we can solve this in the future with the addition of automation to mass photometry, which is a product that we're also bringing to the market this quarter.
All right, thank you for that, Svea. Next question, how often do you need to calibrate the system, and what calibrant is used?
So the calibrant used, as I mentioned earlier, that is a bit up to you. It should be something that you know the mass of, and it's in the range of the measurement of the machine. Typically it's thyroglobulin and AAV. And we recommend to calibrate at least once a day. So whenever something changes, such as the temperature, it's really important that you recalibrate. And also a fresh calibration is especially important if you want to precisely measure the mass. So, but the recommendation is at least once a day.
Okay, and next question we have, this is a pretty good one. Can the Samux MP be used to analyze or for the analysis of other viruses, such as, say, lentivirus or retrovirus?
Unfortunately not. And that the reason is that these are significantly bigger, about 4 times bigger than AAV. And above 80 nanometers in diameter, we are entering a different scattering range. And as we are measuring scattering, we cannot scale the system to that size. So we can only measure AAVs or things of this similar diameter around 25 nanometers or 3.7 megadalton.
All right. And next question we have, one of our audience members would like to know, or they say, when you say particles per mil, what do you mean by particle?
Yeah, that's an important one. So when we optimized the concentration for the measurement, we accounted for every single particle. So that means empty and full AAVs together. Didn't just look at one proportion. So it is the amount of physical particles in the solution, AAV particles.
All right, thank you for that one. Next question we have, an audience member says, besides empty-full ratio, is there a possibility of measuring concentration? Do you also see partial full, which I think you've obviously covered that part as well?
Yeah, I think both are somewhat covered. So with relative concentrations, yes, we are very precise. Absolute concentrations, as in virus titer, we cannot do at this point. And partials, you can see them. You can also use your own analysis pipeline to quantify them. They are just not yet, at this point, part of the quick and easy analysis pipeline. But we're currently investigating it, and hopefully we can launch that as a software update.
All right, and I know this one's probably important for a fair number of our audience. ASPA would like to know if the equipment and software is compliant with 21 CFR Part 11.
It is not at this time point. So we are not GMP compliant at this point. We are looking into this, but this will not be something that comes to the market in 2022. But we are looking into an upgrade version once it's available.
All right, great. Thank you for that. Next question we have, you did kind of just cover part of this, but I thought this was an interesting question, maybe something to talk about. Giuseppe would like to know if the machine can discriminate between AAV and contaminant viruses. And I know you said it couldn't measure other viruses because of the mass range, but if there are viruses close in size, would they be able to discriminate?
If they are exactly the same size as the AAVs, we would not be able to discriminate because we measure the mass. We measure the mass of anything, and it is irrespective of its structure also. So if it's exactly the same size as AAV, as the AAV particles, we would not be able to quantify them. If they are slightly different, then yes. Contaminants, other protein contaminants, they are typically much lower in mass, so they could easily be distinguished. But if they're the exact same weight, I shouldn't say mass, I shouldn't say size, if they are the same mass, we cannot distinguish them, no.
All right, thank you for that. Next question we have, whoops, lost my questions here. Oops, sorry about that. I totally lost my screen. Oh, there we go. Oh, yeah, right. Roxana would like to know if there are any limitations on the buffer used for the assays.
So far, we have tested a lot of different buffers that are commonly used with AAVs, even those that have had amounts of detergents in the buffer, and we have not seen something that we could not yet measure. But there will always be a first. So there are sometimes, if you have really highly scattering species in the buffer, it can happen. But so far, we have measured lots of different buffers typically used for AAV, and we did not have any problems with buffers.
All right, thank you for that. And it looks like we have time for one last question here, which I think is pretty good. Santosh would like to know, how often do you need to change the glass stage?
That's a good point. So we can only measure once at a certain spot on the glass slide, because the molecules would bind to the surface, and then we have to move to a different spot. And we supply consumables, which is the slides, and also a silicon mask, the sample cassette that goes on top of it. This cassette has 6 wells, so we can measure 6 samples per glass slide, and then we would need to change the slide to a new one.
All righty, thank you for that, Svea. And with that, we've come to the end of our webinar. So I'd like to remind everyone that the webinar will be archived on our website at genengnews.com for up to a year. So if you missed any parts of it, you can watch it again, or feel free to forward the link to any of your friends and colleagues, which we always recommend. I'd like to thank Svea for her excellent presentation, and I'd like to thank you, the audience, for your participation and some really great questions that you sent in. And a very special thanks to our sponsor, Refine, for sponsoring this webinar. Hopefully we'll see you again at another Gen webinar in the near future. Goodbye for now. Everyone stay safe and healthy and get your vaccines.